Welcome back. Let's continue. The second point is the selection of the nitrogen source, which depends on plant physiological and salinity conditions. So the second key point I would like to discuss is the impact of the temperature range at the root zone. I will show you an example, a famous example, trial conducted in strawberry by Kafkafi, which had different ratios of nitrate to ammonia. What you see on the four pictures on the left side, these are strawberry plants that were only fed with ammonia. At 10 degrees Celsius, 17, 25 and 32. On the right side, the same strawberry trial, but plants only fed with nitrate. Again at 10, 17, 25 and 32 degrees Celsius. If you look at the root structure at higher temperatures like 32 degrees Celsius, you can see that in the case of ammonium on the left side, all the roots are dead, and consequently also the upper part, the leaves of the strawberry plant. If we look at the nitrate on the right side, you see that the plant is still very productive with a healthy root system and also healthy leaves. So what is the reason for this difference? Once the plant starts producing flowers and fruits, most of the sugars are directed towards the developing fruit, to have bigger and tastier fruits. Only a limited quantity of sugars will be transported to the root to support the root in its functional activities. At higher temperature, the root system gets less efficient with the sugar. There's more root respiration. At the same time, less sugars remain available for the ammonium metabolism that takes place in the root. Ammonium, NH4, is transformed in the root to proteins. And in the first step, ammonium, NH4, is transformed into ammonia, NH3. Ammonia is a very toxic component for plant roots, and it needs immediately to get converted into proteins. This process requires sugars, energy. Now, under these higher temperature conditions, the root system is less efficient with the sugars, and more sugars are getting burned in the root respiration process, leaving less sugars available for this detoxification of ammonia. And that is shown in this study, that at higher root temperatures, when plants are fed with ammonium, the roots will die from ammonium toxicity. Furthermore, ammonium, as shown, competes for the uptake with calcium. That's a direct effect that may limit calcium uptake in the plant. During daytime, most of the water that is absorbed by the plant is used for evaporation, for cooling the leaves. So it goes from the root directly to the leaf. At night, the crop also absorbs water. It's an active process. It's not fueled by evaporation, which just pulls up the water, but it's an active process which involves the root pump functioning. The root is pumping the water up, and this water is mainly going to the fruit, next to the leaves. But this is the water that really helps the fruit to increase in size. A second negative impact of ammonia is that ammonium is reducing this power functioning of the pump. The pump has less power to pump up the water that is directed to the fruit. It also means that less water that contains calcium is pumped to the fruit. So, with more ammonium, the pump is weaker, less calcium gets into the fruit, and therefore the fruit is more susceptible to calcium deficiency. Weaker fruits, increased blossom end rot for those crops that are sensitive to blossom end rot will occur. Very important. Direct impact, ammonium competes for calcium uptake. Secondly, ammonium negatively impacts the pump functioning, and through that way less calcium enters into the fluid. The third point is related to the physiological states of the plant. During vegetative growth, the choice for the nitrogen source is less shown, because the plant is mainly developing leaves. There may be a small reduction in calcium and magnesium uptake, but the plant will not show it to the grower. Once the plant starts developing fruits and flowers, then again the impact is very clear with increased blossom and rod deficiencies. The fourth one, the presence of saline growing conditions. When plants are fed with ammonium, more sodium is being taken up, and at the same time less potassium is taken up in the tissues of muskmelon. If we look at the figure on the left side, we see the two nitrogen sources. The plant fed with nitrate in the light blue color and the one with ammonium in the dark blue color. On the y-axis it shows you the sodium content. It's clearly shown that when ammonium is the predominant nitrogen source, more sodium was found in the leaf blades, the stem and petioles, and in the roots. So why is this happening? Because ammonium has a positive charge and sodium as well. So one may expect that ammonium competes for the uptake with sodium, but this is not the case. And there's an essential step in between this process, and that step is potassium. In fact, it's potassium that regulates the balance between potassium and sodium in the plant. So potassium regulates the sodium selectivity. When there is only ammonium, ammonium will limit the potassium uptake. And therefore less potassium is available to limit the uptake of sodium. So if ammonium takes out of the equation potassium, then sodium has a free ride, so to say. And more sodium will enter the crop because this selectivity, membrane selectivity, that is controlled by potassium is not present anymore. If we go to the right side, where we look at the potassium content, it's clearly shown, as in other studies before, that with nitrate there's a synergistic action with a negative nitrate charge and a positive charge of potassium. They enter the plant together, and therefore more potassium is found in the leaf blades, stems and petioles and roots when plants are fed under the nitrate form compared to the ammonium form. So in general, if we feed plants with potassium nitrate, potassium will compete for the sodium uptake and nitrate will compete for the chloride uptake. So potassium nitrate is a great source under saline growing condition, because in many times salinity is defined by the content of sodium and chloride in the soil. And potassium nitrate helps to keep both ions, chloride and sodium, outside the plant. The fifth point, which is linked to the selection of your nitrogen source, is the water use efficiency. In this study, we are comparing the water use efficiency expressed in grams of plant biomass per liter of water applied or absorbed. And again, we compare an ammonium source with a nitrate source under increasing potassium levels in the nutrient solution. If we look at the x-axis, on the left side we see a very small amount of potassium, 0.1 millimoles, moving to 1 millimole in the center and to 5 millimoles at the right side of the figure. Under potassium deficient conditions, we see that when nitrate is a predominant nitrogen source, the water use efficiency is almost double the amount of ammonia. So that means that when the farmer has limited water resources, he better chooses nitrate nitrogen sources compared to ammonium, because it makes the plant double as efficient with its limited water compared to ammonia. When the potassium content in the nutrient solution increases, the difference is getting smaller between ammonium and nitrate. But still, there is a benefit in water use efficiency when nitrate is being applied under potassium sufficiency levels. And although it may look small, it's still 10 to 15% more. I remember that a grower in Peru developed 5,000 hectares of land in the Los Olmos project. But then he realized that he only had water to irrigate crops for 4,300 hectares. 700 hectares out of this 5,000 had not water to grow crops. 700 hectares out of 5,000 is 10 to 15%. So if this grower could benefit from a nitrate-based program, then he would be able to use this potential area to its maximum. And he could grow crops on 5,000 hectares. Very important how the nitrogen source impacts water use efficiency. Another slide that shows that also photosynthesis is improved with nitrates compared to ammonium per gram. There's more CO2 per shoot per hour, and when it comes to biomass, we can see that there is more biomass produced per plant when nitrate is the predominant nitrogen source compared to ammonia. To summarize, when it comes to the selection of the nitrogen source, nitrate is the preferred nitrogen source. It's non-volatile, easy to handle, no need for incorporation into the soil. Directly available for plant uptake. Nitrate does not need to undergo any transformation, as is the case with urea or ammonia. Nitrate is mobile in the soil, can be directly taken up by the plant, which makes it a highly efficient nitrogen source. Nitrates, with its negative charge, promote the uptake of cations, potassium, calcium, magnesium. There's a synergistic action between the two. It's been confirmed by numerous studies that increasing the ammonium content compared to the nitrate content negatively impacts plant performance, blossom entropy, calcium uptake, fruit quality, fruit yield, and so on. There will be an increased risk of ammonium toxicity at elevated root temperatures when ammonium is the main nitrogen source. The conversion of nitrogen to amino acids, in the case of nitrate, occurs in the leaf. It's a very energy efficient process because the leaves are the source of all the energy. Nitrates help to relieve salinity stress caused by excess of chloride and sulfate. And crops fed with nitrate showed an increase in water use efficiency, photosynthetic capacity, and also biomass production. So we already saw that when it comes to selecting the right potassium source on chloride, do not use chloride containing fertilizers for crops sensitive to chloride. To select from the sulfate and the thiosulfate, those crops sensitive to salinity, avoid any excess of sulfate and chloride. Limit the sulfate supply to the crop demand. In particular, for those crops grown in semi-arid or arid areas, or grown already under saline growing conditions, limit the sulfate content to crop demand. Do not apply any excess, as it will only contribute to further salinization of the rooting soil. Equally important, maintain the right nitrogen balance. In general, 75 to 90% of the total nitrogen demand of the crop should be supplied under the nitrate form, the remainder as ammonia. Remember, with nitrates we have this positive synergy with ions like potassium, calcium, magnesium, and at the same time an antagonism between nitrate and chloride. So with all this information, we can now clearly state that chloride is not desirable, sulfate and thiosulfate under the conditions presented here, and that leaves us to potassium nitrate as the preferred N and K source in plant nutrition. Thank you very much for your attention. It was great being with you today, and I hope you will use all this information to the benefit of the crop. Thank you.